look at this 96 well plate, you will see on the top here we have the reagent blank. The reagent blank, which are these three here, and I hope that you will see them well enough. But in the control set triplicate, you see that the color is much more intense. And in those control wells, you would have the cancer cells and you would have the MTT in there. So it means that the cells are alive, they are viable, and the enzyme is able to convert MTT to this colored compound, and then you have this dark color. In these wells here, they are triplicates, and you have different concentrations of your plant extracts. So you have for sample 1, you have these 3 at different concentrations, sample 2, these 3 at different concentrations, and sample 3, these 3 at different concentrations. If you compare the intensity of the color in the control, you see that for sample 3, even at a high dose, 200 micrograms per ml, you see that the cells are still very much alive. So we will conclude that for this sample here, that the plant extract do not have a high effect to kill the cells, and therefore it would not be something that we want to carry further to do fractionation to see where the activity might be. But in this one here, in sample 2, you see as you increase the concentration of the extract, it almost resembles the reagent blank, which will mean that the cells in this 200 microgram per ml, they're almost all there. So what it means is that the extract has an effect to kill those cancer cells. And therefore, in the case of sample 2, we want to carry it further to do the fractionation with other solvents to see where the activity might be. So we screened, as I said before, we screened about 20 plants with this one cell line. And these are all the plants that I will show you some of them, because the other plants that we looked at, some of them are already investigated by other people, and we found similar results. But those in red are the ones where we found that the crude extract had some effect at a lower dose. Therefore, we want to carry those forward for the fractionation. So there's spermaceti verticillata, you have Flamingia strobelifera, and you have Ficus pumila. And these 3 are the ones that we carried forward to look at the fractionations. This first one, Spermaceti verticillata, is a plant, is a weed, is an invasive species, and in various parts of the world, even in Brazil, is used as a medicinal plant for hemorrhoids and for intestinal parasites. In the Dominican Republic, it is used to treat uterine fibroids. The next plant, Flamingia strobelifera, is also an invasive species, is a pest plant, basically, but it's also used in some parts for medicinal purposes, in Trinidad for kidney stones, and in India for epilepsy and hysteria. Ficus pumila is not a typical medicinal plant in the Caribbean. It's more sometimes used as a hedge, so people will grow them as an ornamental in their yards. But in other parts of the world, they're used for diabetes, hypertension in Japan, and also in China in polyherbal preparations for treatment of breast cancer. So therefore, it's a good indication that we could also use this in our model. So in our first figure here, we actually did a dose response for these 3 plants, which showed activity at lower doses in the crude extract. And what we found is that in methanol extract, for all 3 species, you had relatively low IC50s. IC50 is the inhibitory concentration in which 50% of these cells would have died. So therefore, all of these plots here really is a comparison between the controls and the treated wells. And we found that for Ficus, it actually had the most activity for the crude extract. It was about 23 micrograms per ml. So it means at 23 micrograms per ml, half of the cells in each well would have died. So it meant that that plant is a plant that we probably want to go and look at a little further. So we also did the IC50s for the petroleum ether fraction for these three species, and as we saw in the slide previous, that there's an extraction part in which you do the fractionation. So you have the crude extract, then you mix with water and pet ether, then you have the pet ether extract, then you extract further with ethyl acetate or with butanol. So you have these fractions of these crude extracts, and we also see here that ficus, the pet ether fraction, unlike the fraction we saw before, it had a much higher IC50 value, which will mean that the activity is less in this one than in the previous one. And we see that in Flemingia strobilifera, that the activity is over 300 milligrams per ml. So it indicates us that the activity, the central activity for this plant species probably is not in this fraction. And therefore we can actually start developing an assay-directed approach. In this one we have the ethyl acetate fractions, and we see that all of them over 200, and it indicates to us that the activity that we see previously most likely would not be here. And of course, in the butanol extract, you see only Ficus pumila have an activity of 26 micrograms per ml, which would suggest that Ficus pumila most likely would have activity in those fractions. Therefore, it limits the amount of chemicals that we have to search for when we do fractionation. And the chloroform extract, again, we see that for Ficus pumila is 23 micrograms per ml. So therefore, Ficus pumila, which is the creeping vine plant, probably has potential for us to go even further to determine what is the active compound, the chemical that we can find over there. So just to review, we see that for these plants, that actually Ficus pumila is the one which has the most activity, both in the crude extract and in the other. So you see that the chloroform extract, the butanol extract, the activity is the lowest at 26 and 23. So therefore that will give us an indication that in the future, when we go forward, that these are where we should probably be focusing on. So in conclusion, I know that we probably in the middle of a siesta now, that we did look at about 20 plants. We saw that there were 3 of them which we wanted to carry further to do fractionation. And of those, we saw that the highest activity was found in the chloroform and butanol extract of the Ficus pumila. Similar studies were done to determine, well, not similar studies, but phytochemical studies were done on some of these plants, and they have shown that they contain certain compounds. So we can actually start having an idea what kind of compounds that we should be looking at. And there are some glycosides, and there are also some flavonoids and beta-cetol-steroid glycosides. And some of these compounds have already been shown to have activity against bacteria and fungus. The plant that we found that with the most activity, Ficus pumila, it contains at least 5 flavonoid glycosides, which include a compound called rutin. And rutin was found in another plant, and they have shown that rutin actually prevents the carcinogen-induced breakage of DNA in a rat model. They have also shown that rutin, which is contained in this plant, is a polyphenolic compound, and it has activity against another cell line, another leukemia cell line, by inducing programmed cell death or apoptosis. And therefore we suggest that the rutin that is found in Ficus pumila in this one might be responsible for some of the activity that we see in the plant. But we are not too sure, but so it means that we have to go forward and we have to start to isolate and identify the compound in Ficus that we think might be responsible for the activity. And other studies have shown that besides these flavonoids, we also have these sequestered terpenoid glycosides in the fruit of the plant, and they have shown that these terpenoids, that they have cytotoxic activity against human leukemia cell line as well. and they're actually 3 times more potent than etoposide. So we suggest, actually this is preliminary data, it means that we have to go further from the fractions to actually do some chromatography and isolation of compounds to determine what the actual active ingredient might be for providing activity against the cell line. We suggest too that probably it might be some antioxidant, because we know that flavonoids they have high antioxidant properties, and it could probably be that besides affecting cell growth and different events in the cell cycle, they may actually be interacting with intracellular mechanism to produce antioxidant effects. So therefore, there's a lot of work still to be done. We have only looked at 20 plants. We have looked at 1 cell line. Like I said before in Trinidad, the major cancers are breast cancer, prostate cancer, and colorectal cancer. So it might be that we have to develop those cell lines as well and to move forward. And I want to thank these people there. So, alright, so in the lab, the technician that I work with is Juan, and he has done most of the work in the lab to isolate and to do the fractionation and to do all of the work with the cell line. And Prof. Tilan and Prof. Knott, they were the ones who actually initiated the work, and I came on after. And Ms. Jasmine Comon is the person in the National Herbarium who would look at the plants and make sure that we have the right plan before we go forward. And of course, in the Caribbean we have a problem with funding, but at the university there's a small fund where we can actually get monies to do bits of research. But this, as I said, is very preliminary, and therefore we have to go further with it. So thank you very much.